This is a Pinnacle how-to video demonstrating the targeted quantitation workflow. The purpose of this video is to provide an overview of setting up the workflow, processing data, and then interpreting the results. And we'll cover the general concepts of the workflow. The specific features and attributes of the targeted quantitation workflow are covered by specific or topic-specific videos that can be also accessed in the same location. So when you first open up Pinnacle, this is the view that you would see. And so the user would go to this preset workflow, drop this down, and then select the targeted quantitation. Once you select the targeted quantitation, all of the features and attributes that the user would need to set are contained over here on the right-hand side. Specifically, you would start at the top and work your way down, setting up the specific attributes to performing the data processing. So first you would simply browse to find as many of your files as possible, so you can just simply click on the add files and then go to the specific directory and grab as many of the files as you need and then bring them in and hit done. The second thing and the most important attribute for the targeted quantitation is defining your targeted compound list. And this can be done in a number of different ways. So you have the option of going to a custom sort of that list of peptides and their attributes that were pasted into an Excel grid that you can just simply copy and paste and import into Pinnacle. You can go to a spectral library, and this is in reference to Proteome Discoverer. So you can grab the MSF files from Proteome Discoverer, or you can grab the crystal libraries, and that's a node that's within Proteome Discoverer that you can grab and bring or import into Pinnacle. You can also do the same thing with mascot DAT files, and then lastly CSV files. So again, a customized or user list that you can then bring in. And of course, these are with both pasted from Excel as well as the CSV files. These provide you an opportunity to import results that were created from another searching algorithm. And so the benefit of the spectral library is just simply you're reading in or providing all possible proteins and peptides information into Pinnacle that it will then use to process all of the raw files that you've provided. So in addition to the protein and peptide list, Pinnacle also reads in the charge states for both precursors and products, the product ion distribution, which is absolutely key for scoring and increasing the confidence in evaluating your data. It reads in all of your PTMs that were identified during the search parameters. And finally, it reads in the retention time for that particular peptide. And depending on the consistency between the data acquisition for generating the spectral library and for the data that you're going to be using to process it, you can set up retention time windows from which to extract and integrate and quantitate the data. So the thing that so these particular attributes of reading in the targeted compound list dramatically expedite the data processing, as well as simplifying the searching. That is, the user does not have to then pick and choose which proteins and peptides from which to do the quantitation on, nor does the user have to select a list of possible either static or dynamic modifications. That's already been done during the initial search to create the spectral libraries. And again, this increases the how fast the data processing happens. Once that is done, you can go down into the next section, which is the file reading. This is simply setting the mass tolerance values for MS and MS2. You would go into peak processing, normalization strategies, scoring, and then layout. And again, all of these individual parameters have their own videos associated with them. So once you've brought in the raw files established, as well as the targeted or the spectral library, and then established the searching parameters, you would just simply hit apply, and that begins the data processing. I've already processed some data, and this is a comparative study, relative quantitation. And it's a complex system, so you can see that we've processed across 5 raw files about 2,500 proteins and there's about 10,000 peptides. And so processing these 5 raw files from a QExactive took about 15 minutes. And the data that we're going to process, there are 3 different groups. And within the groups, and we can resize this here, within the specific groups we can look at the file areas or the files. We have 2 for one group, 1 raw file for the middle group, and then the last 2 belong to, again, the 3rd different group. And the order that you see here, and of course the grouping has already been covered in a video, but this will then show the arrangement or the order for our gel plots. And so once you've processed the data, you have this particular layout, which is the results table showing a comprehensive overview of all results. So you don't have to pick and choose which protein to look at specific results. You get everything in this particular table. You have a chromatographic view, and then you have a mass spectrum view that you can look at either the MS or the MS2. And as the default layout that you can set up simply showcases both a gel view, which is shown here, from the file. So each one of these individual bars or gels represents the response or the average response for all peptides for the individual raw files. And again, the first 2 would represent one group, the 3rd one represents its own group, and then the last 2 would represent the 3rd group in the study. Then you also have a group ratio view, in which this is the average response for the all raw files associated with each individual group. And so what this provides me is I can very quickly, and the last thing is you also would see the peptide count for this particular protein. So you gain some perspective as to how well the protein is covered and what the response is. And as I just simply scroll through this, I can very quickly look to see how the proteins are behaving across my different groups, both from the individual files. Are they consistent? And again, we would expect so because these are technical replicates, 2 technical replicates for each group except for the middle one, as well as the average response showed here in the group ratio. And again, we can just sort of scroll through this, and we can simply look at the responses. The additional views that you would have are shown here in the chromatographic view. And again, as we're scrolling through this, we see the different responses of our particular proteins. And we can simply grab some that look kind of interesting. And again, so this one shows that the response levels are sort of decreasing across the files. When we click on it, again, we've got 14 peptides that are associated with this particular protein. We can also expand our view here, and now we can not only see at the protein level, but we have all of our peptides as well. And the table that you can simply scroll through will give you the maximum file area, molecular weight, modifications, retention time. So you'll get all of that information for this specific peptide. But you have the peptide sequence, you have its response factors. Do they all show similar responses, both from the individual files as well as the group? And it gives you, of course, the length. In the chromatographic view, we can simply, again, there are 5 different raw files. We show all peptides that are eluting in this particular time window. And we can now set this to simply see what's going on per peptide. And if we simply grab a particular peptide, open it up, or highlight it, we can now see the individual response across all of our individual raw files for this particular peptide. We can, of course, see where the MS2s have been triggered. We click on it, we can get some estimation of how well things are matching up. And we can also then look at the MS response for our particular peptide and zoom in to evaluate the mass accuracies. This ends this particular video.